Glycolysis is the major pathway of glucose metabolism and occurs in the cytosol of all cells. It developed before oxygen was widely available on Earth and produced pyruvate and ATP from starting glucose. While this is not the highest possible yield mechanism for ATP synthesis, it does produce some, and the pyruvate produced can be used to go into fermentation, which can also generate some ATP. Under aerobic conditions, the pyruvate produced moves into the citric acid cycle and electron transport chain, where it yields approximately 32 moles of ATP per mole of glucose, while oxidation of glucose under anaerobic conditions yields only 2 moles of ATP.
Glycolysis occurs in two stages. The first stage of glycolysis consists of glucose being trapped in the cell and modified with cleavage into phosphorylated 3-carbon compounds. ATP is invested in this stage so that two 3-carbon glyceraldehyde 3-phosphates can be generated from one 6-carbon glucose molecule. Then in the second stage, the oxidation of the 3-carbon compounds to pyruvate with the generation of 2 molecules of ATP per pyruvate molecule occurs, which results in a net production of 2 molecules of ATP.
Among the 10 reactions in glycolysis, there are only 3 regulated steps. The first step, the third step, and the 10th, also known as the last step. Regulation in this context means that the reaction is irreversible. The third step of glycolysis, which is the second irreversible step, is catalyzed by the most important regulator of glycolysis, the allosteric enzyme called phosphofructokinase, abbreviated as PFK. PFK catalyzes the reaction of fructose 6-phosphate into fructose 1,6-biphosphate. In this step, ATP is transformed into ADP. PFK is allosterically inhibited by ATP and also allosterically stimulated by AMP. Therefore, a lower concentration of ATP increases the activity of PFK. This regulation makes sense because it is when the cell's energy decreases that glycolysis is especially needed.
A non-regulated step in glycolysis consists of the reaction from glucose 6-phosphate G6P to fructose 6-phosphate F6P. This reversible reaction is catalyzed by phosphoglucose isomerase, which falls under the type of enzyme that converts a molecule to another isomeric form of itself through intermolecular bond rearrangements. G6P is first converted into its open chain form, resulting in an aldehyde on the first carbon. The enzyme then isomerically forms fructose through the rearrangement of bonds to produce a ketone on carbon 2 and an OH group on carbon 1. Finally, this open chain form of glucose is converted back into the ring structure of fructose 6-phosphate, F6P. Another non-regulated step in glycolysis is the cleavage of fructose 1,6-bisphosphate into dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, GAP. The reaction is reversible and catalyzed by the enzyme aldolase. Both these steps are not regulated because the reactants and products are closer to equilibrium compared to the regulated steps. There is essentially no point in regulating the reaction if the reaction is already close to equilibrium because the forward and reverse reactions occur at the same rate.